Hello Professor, I am Mubashir Raza. The year of my registration is Spring 2022. I am master student in molecular biology and human genetics and my advisor is Professor Ingrid Value. Here is the checklist of my progress report. In current semester I have registered 6 courses which contains the credit hours of 6. Timeline. I reached on 25th April in Sachi University. In April and May, I have learned different techniques in my lab. These are cell culture, pipetting, sample preparation and genotyping of mice, restraint stress and animal behavior, open field test, mice handling. In June, I am learning about Western blot. In this experiment, I learned about the culturing of HT22 cells, and here is the confluency of HT22 cells after 24 hours, after 48 hours, and at 4 days. I have learned about the genotyping. I have performed ATF3 genotyping of the mice, and I got the result of wild type mice and heterozygous mice. In this experiment, I learned how to give restraint stress to the mice and especially ATF3 mice. I also learned about how to handle the mice during animal behaviors test as well as during drugs injection. I learned about the Western blot and how to analyze the proteins. For this, for the sleep deprivation mice. Future direction. I have a project. The title of the project is the effect of Litsi cubiva on pathology of Alzheimer's disease in vitro and vivo studies. Alzheimer's disease. Alzheimer's disease is a neurodegenerative disease. It has two pathological hallmarks. One is extracellular deposition of amyloid beta and second is intracellular deposition of tau neurofibrillary tangles, which causes the shrinkage of hippocampus, shrinkage of cerebral cortex and enlargement of the ventricles in the brain of the Alzheimer patient. Litsi cubiva is a medicinal plant. It has antioxidative and anti-inflammatory effects. So for this purpose, I am going to use LC as a medicinal drug for Alzheimer's disease. Hypothesis of my study: LC can improve the pathology of AD in vitro and vivo studies. In vitro studies, I have selected the cells HT22 cells, and for in vivo studies, I have selected 3xTg AD mice. Aim of my studies. So specific aim 1, in first experiment, I will investigate the effect of Litsi cubiva in HT22 cells. For this experiment, I have 3 group of cells. One is control, second is sham, third one is LC. I will treat the cells with different concentrations of LC, and after that I will perform the MTT assay and ATP assay to check the cell viability. The expected result of this experiment is LC will not show any toxic effect on HT22 cells. Specific aim 1, in second experiment, I have different 7 groups of the cells treated with LC1 and 2. First I will check the MTT assay and ATP assay, then I will perform the Western blot, immunocytochemistry and ELISA kit to check the markers of inflammatory and oxidative stress. Expected result of this study is LC will reduce amyloid beta toxicity by inflammatory and oxidative stress markers. Specific aim 2 is about the study of effect of LC on learning and memory of 3xTg AD mice. For this I have 5 group of mice treated with LC1 and LC2, and then I will perform the behavior test for each group. First I will perform the open field test for locomotor activity, T-maze for working memory, Morris-Water maze for spatial memory, trace field conditioning for learning and memory. And expected result of the study is LC will improve the learning and memory deficit in 3xTg AD mice. In aim 3, I will determine the mechanism of LC that regulates the learning and memory in 3xTG AD mice. For this study, I have 5 groups of mice. Each consists of the 36 mice having age of 6 months. First, I will perform the behavior test, then I will sacrifice the mice and collect the protein from hippocampus, prefrontal cortex and amygdala, and collected the brain tissues, and then perform the long-term potentiation, ELISA test, Western blot and immunohistochemistry to find the inflammatory markers, oxidative stress markers and microglial activity. And the expected result of my study is LC will improve the learning and memory deficit by inflammatory, oxidative stress and microglial activity markers in 3xTG AD mice. Along with this project, I have learned about the different techniques. I will learn about the different techniques. These are Morris water maze, T maze, trace field conditioning, tail flick test, and pin prick test, elevated plus maze, passive avoidance test. I will learn about the techniques like immunohistochemistry, Western blot, and qPCR. And I will in upcoming semester I will selected the courses of 8 credit hours to complete my graduation criteria. That's the end of my presentation here. Thank you so much.